This is a review of urinary system anatomy. Here you can see the organs of the urinary system. This system includes right and left kidneys, right and left ureters, a single urinary bladder, and a single urethra. The kidneys are partially protected by the inferiormost ribs and are retroperitoneal. The ureters, urinary bladder, and urethra form the urinary tract. In this cross-sectional view, you can see that there are two layers of fat surrounding each kidney. The tissue that forms the renal or fibrous capsule is a layer of irregular, dense connective tissue. Superficial to that is the adipose capsule, or perinephric fat, which is enclosed by the renal fascia. Superficial to the renal fascia is another layer of fat, the paranephric fat. This layer is deep to the parietal peritoneum, making each kidney retroperitoneal. Each kidney has a hilum, the point at which the renal artery, renal vein, and ureter connect to the kidney. In this coronal section, you can see the internal organization of the kidney. The ureter is continuous with the renal pelvis, which at its outer curvature collects urine from the major and minor calyces. Each minor calyx is a funnel-shaped structure that collects urine from a single renal pyramid. Major calyces are formed when minor calyces join together. The renal sinus is the region at the hilum that contains the renal pelvis and major blood vessels. You can think of the renal sinus as a hollow indentation at the hilum of the kidney. It is surrounded by the region called the renal medulla, which is deep to the renal cortex. As with other organs, the cortex is the outer region and the medulla is the inner region. The renal medulla has distinct renal pyramids, separated from each other by renal columns. The renal column is actually a continuation of the tissue of the renal cortex, so that cortex plus column surrounds each pyramid. This cortical tissue contains the main portions of the nephrons. Each pyramid contains some part of the nephrons, but consists mainly of collecting ducts, which drain the forming urine towards the minor calyces. The tip, or papilla, of each pyramid is surrounded by the minor calyx, so that urine flows from the collecting ducts in the pyramids into papillary ducts in the papillae, then into the minor calyces. In humans, no further adjustments to urine composition or volume can be made after it leaves the collecting ducts. Here we see the microscopic anatomical organization of the kidney. There are three principal parts: the renal corpuscle, the renal tubule, and the collecting system. The renal corpuscles are always in the cortical tissue, renal cortex or renal column. The renal tubule is primarily in the cortical tissue, but has a specific part, the nephron loop, that can invaginate into the renal medulla. The collecting system begins with collecting tubules, which attach to each nephron. These join into larger collecting ducts, which begin in the cortex and end by draining into the papillary ducts within the renal papilla. In this image, You can see that some nephrons are contained almost entirely within the cortex. These are cortical nephrons. The nephrons that originate at the corticomedullary junction typically have longer nephron loops that dive deep into the medulla. These are called juxtamedullary nephrons. About 20% of human nephrons are juxtamedullary. They play a crucial role in regulating the amount of water reabsorbed as fluid flows through the neighboring collecting ducts. These micrographs allow you to compare the histological appearance of the renal cortex and renal medulla. Note the presence of the renal corpuscle only in the cortical tissue. The renal medulla, sectioned through a pyramid, shows it to be mostly a cluster of ducts and blood vessels. This diagram shows the structures that make up the renal corpuscle. The boundary of each corpuscle is the glomerular capsule. A balloon-like structure composed of simple squamous epithelial cells that is continuous with the renal tubule. Each corpuscle contains about 50 capillaries, collectively referred to as the glomerulus, a word that means little ball. Each glomerular capillary is individually enclosed in a layer of cells called podocytes, forming the visceral layer of the glomerular capsule. Blood flows from the afferent arteriole into the glomerular capillaries and is drained into the efferent arteriole. Specialized cells within the afferent arteriole, called granular cells, or juxtaglomerular cells, are connected to adjacent cells of the distal convoluted tubule called macula densa cells. The granular cells and macula densa cells collectively form the juxtaglomerular, or JG, apparatus, which plays an important role in regulating blood pressure and urine composition and volume. Glomerular filtration is the process by which water and solutes within the plasma are forced into the space of Bowman's capsule. This fluid, called filtrate, will flow into the proximal convoluted tubule and will then be called tubular fluid. In this image, we see the renal vasculature. The renal artery divides into segmental arteries in the region of the renal hilum. Segmental arteries give rise to interlobar arteries, which run between the renal pyramids. At the corticomedullary junction, interlobar arteries end by branching into arcuate arteries, which arch over the pyramids. The arcuate vessels are found at the boundary between the medulla and the cortex. Each arcuate artery gives off numerous interlobular arteries, which penetrate the renal cortex. Each interlobular artery has multiple afferent arterioles, each of which supplies a single glomerulus. Blood leaving each glomerulus drains into an efferent arteriole, which empties into a second capillary, either a peritubular capillary within the cortex or a vasa recta associated with a nephron loop. These capillaries drain into interlobular veins, to arcuate veins, to interlobar veins, and then into the renal vein. The arteries and veins with the same names run side by side. The renal vasculature forms a portal system, the renal portal system. Recall that a portal system includes two directly connected capillary beds. In the renal portal system, blood from the glomerular capillaries is drained through arterioles, not veins, into the peritubular capillaries before returning to the typical venous drainage. In this image, you can see the change in terminology for the fluid formed by the urinary system. from filtrate in the capsular space to tubular fluid until it leaves the collecting duct and finally to urine. This image allows you to see the continuity of a single nephron. Glomerular filtration occurs within the renal corpuscle. About 180 liters of filtrate is formed every 24 hours. Once the fluid enters the renal tubule, tubular reabsorption and tubular secretion occur. This involves moving substances between the tubular fluid and the blood within the peritubular capillaries and vasa recta. About 178 to 179 liters of fluid is reabsorbed into these capillaries every 24 hours, leaving only about 1 to 2 liters of urine formed in a day. In these cross-sectional views of a ureter, you can see the layers of the mucous membrane. The epithelium found here is unique to the urinary system, a transitional epithelium, which is a stratified epithelium in which the cells are unusually stretchy. This tissue type is also found in the urinary bladder. The muscularis layer is visceral smooth muscle, which can sustain contractions in waves, a process called peristalsis, to propel urine from the renal pelvis all the way through the ureteral opening and into the urinary bladder. The features of the urinary bladder include a highly distensible upper region with a stable base called the trigone. The corners of the trigone are marked by the 3 openings or orifices, right and left ureteral orifices and urethral orifice. The surface of the trigone is smooth, unlike the wall of the rest of the urinary bladder, which is folded into rugae or mucosal folds, similar to the stomach. The smooth muscle of the urinary bladder is called the detrusor muscle. It is formed in separate small bundles rather than one continuous sheet, so that when the bladder is full, the bundles can be stretched away from each other without overstretching the muscle fibers themselves. The muscularis surrounding the urethral orifice has a specific circular arrangement and forms the internal urethral sphincter. Typical for sphincters, the muscle stays contracted most of the time, relaxing only to permit passage of urine out of the bladder, ordinarily only when the bladder contracts. These images highlight similarities in and differences between female and male urinary tracts. The external urethral sphincter, embedded in a region of the pelvic floor called the urogenital diaphragm, is composed of skeletal muscle. As with the internal sphincter, the default state of this muscle is to remain contracted, although that response is developed only with the conditioning of toilet training. In babies, and in certain cases of paralysis, the external sphincter does not contract, and the bladder drains reflexively whenever it fills to a certain degree. The female urethra is short, ending at the external urethral orifice within the space known as the vestibule between the labia minora. The male urethra conducts not only urine, but also semen. As the urethra begins on the inferior aspect of the urinary bladder, it immediately enters the prostate gland. This is the prostatic urethra. The prostate rests on the urogenital diaphragm. Again, part of the pelvic floor containing skeletal muscle. The short region of the urethra that passes through this structure is the membranous urethra. The last, longest region is the spongy or penile urethra. This ends at the external urethral orifice. This concludes the review of the urinary system.